Good afternoon, everyone. Really? Thank you, Mary. Good afternoon, everyone. You know, if I were really smart, I would have brought in some kind of trombone and some cheerleaders or something, because this is the equivalent of Friday night football. Or, San Francisco 49ers football in the scientific world. Good. Hello, everyone, and welcome to the sixth annual Postdoc Slam competition at UCSF. All right, so that's not for me, that's really for you. I am Nikkei Blake, and I have the honor of serving as your dean of the graduate division here at UCSF. That's at least one of my roles. So Grad Slam is really about celebrating postdocs. It is part of Postdoc Appreciation Week, which happens the third week of September in every year. So, in light of that, if you are a postdoc and in the room, please stand. Don't be shy, don't be shy, don't be shy. There you go, there you go. All right, no, stay standing. All right. If you are in the vicinity of a postdoc, just give them a high five and say thank you. If you're in any vicinity of a postdoc. All right. All right. Well, welcome, everyone. At UCSF, when we convene at significant gatherings, we take the time to recognize through an official declaration that the public event is occurring on territory originally inhabited by indigenous communities. As we gather here in person on our campus in San Francisco, we want to acknowledge the Ramaytush Ohlone people, who are the traditional custodians of this land. We pay our respect to the Ramaytush elders, past, present, and future, who call this place, the land that UCSF sits upon, their home. We are proud to continue the tradition of coming together and growing as a community. We thank the Ramaytush Ohlone community for their stewardship and support, and we look forward to strengthening our ties as we acknowledge our relationships of mutual respect and understanding. All right, thank you. Like I said, Postdoc Slam is part of Postdoc Appreciation Week. Postdocs, have you been having a little bit of fun this week? No? No fun? Postdocs are not allowed to have fun. Is that what? Not allowed to have fun. Yeah, I understand there's some faculty in the room, and we can't acknowledge that we have fun. But it is just the beginning. Postdoc Slam actually benefits our entire community. First of all, it benefits our competitors, and we want to take a moment just to acknowledge our competitors right off the bat. So will all of you please rise? Yes. Well done. Thank you very much. We hope that the skills that the contestants have sharpened in preparing and practicing their talks today, and their poise will have gained, they will have gained from engaging in the challenging communication exercises, will serve them well, no matter where their career paths take them. Excuse me. The last few years have only reinforced that how important it is for us as scientists to communicate well. No one has to remind us about what happened during COVID and all the misinformation that perpetuated across the country in light of a vaccine for the virus. And part of our responsibility as scientists is really to communicate well to the taxpayers who largely support the research that you conduct. So Postdoc Slam benefits those guys because they learn a lot, but it also benefits the rest of us because it gives us just a glimpse into the fascinating research that is happening at UCSF every single day. This is my second, I think, postdoc slam, my fifth slam in general, but my second postdoc. And I got to tell you, every single time I come in this room and have the opportunity to cheer, it is just an amazing event. So I am looking forward to hearing those guys present the work that they are doing. So thank you all for all your hard work in preparing, and we are all excited about hearing about your work. I want to take a moment to introduce someone who is brand new, but very special. Dr. Shaquita Allen. Shaquita, can you just stand up for me? So Shaquita is our brand new postdoc dean. You will all get to know her very well. Some of you are already getting to know her. And we are excited to have Shaquita on the team. You've managed to survive without a postdoc dean for about 2 years. So it's taken a minute, but we are thrilled to have Shaquita in the house live and in living color. Shaquita brings a wealth of leadership experiences in academic and corporate settings to the role, and we are just excited to see what she will do with your help over the next months and years. All right, now, normally I just slide into the show right now, but this year, you have a special treat. So this year we decided that no one wants to hear from a dean, but boy, would you want to hear from a former EVCP. So we invited Dr. Dan Lowenstein to MC the event today. Anybody know who that is? No? No one has any idea who that is? All right. Well, let me just tell you, if you think about your best football, baseball, soccer commentator, no comparison to what you were about to receive. No pressure, Dan. Absolutely no pressure at all. So Dan is an esteemed professor of neurology in the School of Medicine, an expert in the genetics of epilepsy, and former EVCP from 2015 to 2022. But Dan is also a longtime fan of the Slam. He is a veteran of the judging panel. Every time we ask Dan to volunteer, he says yes. He is incapable of saying no to the Slam, quite frankly. So I am thrilled to invite Dan to the stage now to take you through the process. Dan.
Thanks, Nikay. Thank you. Thank you, Nikay. My goodness, I can't think of anything that exemplifies what this university, what this institution is all about. You know, the people of society expect us to generate new knowledge so that we can lessen the suffering in the world. That's the bottom line. And every single person in this room is involved in that. And that's what makes this one of the most exciting events of the year. So it's a pleasure. Thanks for the invitation to be MC. I'm kind of getting my energy up here to figure out how to meet your expectations. But basically, today you're going to hear from 10 of UCSF's over 1,000 postdocs. And the postdocs who entered into the Postdoc Slam contest by submitting their talks on video, and a group of screening judges that included faculty, staff, and alums reviewed these videos to choose our 10 finalists. So you can just imagine, with the anxiety that they hold in preparing for this, they've been working really hard to make this as engaging as possible for all of you. And of course, the prize money doesn't hurt as well. For the competition, the grand prize is $4,000. Second prize is $2,000, and third prize is $1,000. And all of you in the audience, both in person and those of you who are watching remotely, you get to also pick a winner, and that's deciding on the People's Choice Award, which is worth $750. So you'll have a few minutes to cast your vote online after all the finalists have given their presentations, and I'll give you some more details on that later. Okay, the other way the in-person audience can participate, the folks here in the room, is in our famous trivia contest. Between each speaker, I'll ask you all a question, and the live audience member who gets it correct will win a very special prize. Okay, so we'll get to that too. Now, I know you're anxious to hear the students' talks, but before we started with the presentations, I want you to introduce you to a very, very impressive panel of judges. Judges, if you don't mind, just stand briefly as I introduce you one by one. Curtis Chin is a San Francisco Unified School District science teacher, teaching at Galileo High School since '96. He's also a longtime partner of UCSF's Science and Health Education Project and the host of the Infatuasian podcast. So welcome, Curtis. Hal Collard, UCSF's Vice Chancellor for Research and Professor of Medicine and Health Policy. There you go. Juan Ha is UCSF's Vice Chancellor of Communications. Audra Johnson is a medical science liaison at Genentech and UCSF postdoc alum, where she participated in the ARACTA postdoctoral program. Nice to see you. And then Helen Wilsey is an assistant professor of psychiatry at UCSF, an investigator with the Chan Zuckerberg Biohub Investigator Program, and also a UCSF postdoc alum. Welcome. Okay, so you've got I know this, you've got a really hard task in front of you, but thank you for being here and judging this important competition. So the judges are going to be looking for these basic qualities in the presentations. I think we have a slide for that. Did the presentation help you understand the research? Was the topic and its significance communicated using language appropriate to an educated but non-specialist audience? And did the presentation make you want to know more about the research? And each talk will be timed, carefully timed, and any presentation that goes over 3 minutes will be disqualified. But don't worry, there's not going to be any screw-ups on this because our official timer is the UCSF registrar, Doug Carlson. And not only does he look after all of your grades and so forth, he's the official timer for the Cal sports teams. So he's a real pro at this. And that's actually really true, by the way. Okay, and just want to let you know, please silence your cell phones. We want to eliminate any distractions during the talk. So also, please, no talking, no texting, no flash photography during the postdoc presentations, no applauding or yelling out afterwards. No, just kidding about that. Okay, so judges, you all ready? Okay. Finalists, you guys, you ready? Okay. Everybody else ready? Okay, so here we go. So the finalists drew numbers to determine the order in which they'll speak. So our first speaker is Tyler Hike. Faculty members Zev Gartner and Ophir Klein. The talk title, How the Gut Got Its Spots.
Your intestine is filled with fingers. Now, that might not be what you wanted to hear right now at the beginning of the slam, but your intestine is filled with millions of microscopic finger-like structures called villi. These are essential for nutrient absorption, but they can become damaged and fall apart, sometimes even leading to lifelong digestive complications. One way to treat this would be to grow intestinal tissues in the lab to transplant into patients. But in order to do this, we first need their blueprints. These blueprints are in the embryo where these structures are first built. And a major focus of my research is to understand how the embryo builds tissue structure, like villi, so that we can build it ourselves. Now, to begin unlocking these secrets, I've developed ways to grow the intestines outside of the embryo and in a dish where we can use high-powered microscope to watch as they develop and change shape over long periods of time. Now, if you were an engineer tasked with building structures like these villi, you might start by making a scaffold that looks like fingers and then dumping some cells on top. In the embryo, however, the cells are the engineers, and they've decided on quite a different way of doing things. And it begins with a series of microscopic spots that appears along the whole length of the intestine. So how did the gut get these spots, and what did they do? Well, one way to learn how things work is to take them apart. So I broke down the intestine into its individual component parts, the individual cells, and watched as these cells rebuilt these structures. So they reorganized back to form these aggregates that are highly cohesive and have a high surface tension. Now, you might be familiar with something else with these same physical properties, water. And because of these properties, water will also reorganize to form droplets, just like on the windshield of your car. So maybe these cells were behaving like water somehow. And then I had a penicillin moment. I had left some of these intestines growing in the lab over a long holiday weekend. And when I came back, I found that not only had these spots formed, but they had fused together, just like two droplets of water would if they came into contact with each other. So I've unlocked the first steps through which the embryo builds these intestinal villi. And it does so in the same way that water makes droplets. These cells minimize their energy to generate these droplet-like aggregates, and in doing so, they generate a surface tension that folds the inner lining of the intestine to initiate where these finger-like protrusions, these villi, will grow from. So we can now use this knowledge to begin engineering these structures ourselves in the lab. But beyond this, we can now apply this kind of framework to better understand these complex living systems like our tissues by comparing them to the better understood physics of the non-living world, things that we encounter every day like water. And in doing so, we can learn how tissues are built so we can now build them ourselves. Thank you.
Thank you, Tyler. Okay, judges, you can go at it here, and let's go for the trivia contest. So today's loose theme is science and pop culture. And for those of you who are watching remotely, this will be like watching Jeopardy. In other words, sorry, no prizes for you. But we hope you'll have fun guessing at the answers anyway. But for our live audience, please raise your hand. Raise your hand if you know the answer. And obviously, no Googling or whatever to find the answer on your cell phone. We don't have audience mics today, so Yana Avila, our new Chief of Staff in the Grad Division and Student Academic Affairs, she's going to point to the first raised hand she sees, and if that's you, say the answer loudly. We don't have any mics floating around, so just holler out the answer. If you get the answer, we have a goodie bag for you. I want to thank Campus Life Services for generously donating our trivia prize swag. And let's not get too overly competitive about this. Like every year, you know, there's little arguments here. The way I raise my... Forget that stuff. Just whatever. Okay, so here we go. The first question. What was... Are we all ready here? Yeah. What was the full name of... Oh, did someone raise their hand? Okay, we got... Yeah, what was the full name of the murderous AI in 2001, A Space Odyssey? Hal, can you add anything for a couple other people? That is the correct answer, HAL 9000. Okay.
Open the pod bay doors, Hal.
I'm sorry, Dave. I'm afraid I can't do that.
Okay, so HAL, also known as the heuristically programmed algorithmic computer, was the main antagonist in Arthur Clarke's Space Odyssey series, and he first made his appearance in Stanley Kubrick's 1968 film. And while part of HAL's hardware is shown towards the end of the film, he's mostly depicted as a camera lens containing a red and yellow dot. In the film, HAL becomes operational in January of 1992. That was years after the film, at the HAL laboratories in Urbana, Illinois, and was responsible for maintaining the Discovery 1 spacecraft systems during the interplanetary mission to Jupiter. Interesting stuff here. Speaking of vocalizing, HAL was voiced by Douglas Rain, who, according to Kubrick, quote, had the kind of bland mid-Atlantic accent we felt that was right for the part. And one last piece of trivia. The, quote, bland accent helped HAL achieve the distinction of the 13th greatest film villain in American Film Institute's 100 years, 100 heroes and villains. Fittingly, it's reported that Anthony Hopkins drew in part on HAL 9000 for his performance as Hannibal Lecter in the 1991 film, The Silence of the Lambs. There you go. Okay, judges, are we good? Okay. All right, then we are ready for our, yes, Dave, thank you. Okay, now for our second speaker, Amanda McQuaid, a faculty member, Martin Campman, and the talk title is Speaking to Cells. Amanda.
Everybody talks, everybody talks. Everybody talks, everybody talks. Everybody talks, everybody talks.
When you get sick, your immune system responds. And this isn't just in response to colds or viruses. The immune system also responds to brain diseases like dementia. In fact, the majority of genetic risk factors for dementia influence immune responses, suggesting that these cells hold great power over brain health. Unfortunately, in the case of dementia, they seem to be using these powers for evil. We've caught these cells spreading messages of panic across the brain and tearing their neighboring cells apart. And I just want to tell them to stop. But obviously I can't speak to cells, or at least not yet. It turns out we do know a fair bit about how cells communicate. Their language is encoded in DNA. The DNA is naturally made up of segments called genes, which I equate to words or phrases. When a cell wants to mount an immune response, it reads a specific gene from the DNA code. In the case of dementia, they seem to be reading panic and attack everything. But I want to see if there are words that we can speak to these cells that will help them calm down and restore health in the brain. To do this, I'm using a technology called CRISPR activation that lets me find a specific part of the DNA in the lab and read just that word. Then I can see how the cell responds. Does this make you feel like shouting fire in a burning building? After looking through all of the DNA, I now have a fairly good idea of how to speak to cells. I figured out the words that seem to mean panic, other words that mean restore peace, and I'm even able to form more complex phrases like protect the healthy cells, but keep an eye out for intruders. So it's been very exciting to learn how cells speak about dementia, but now I want to take this back into disease. Right now I'm working on coaching these immune cells in a 3D brain environment to see if just shouting restore peace is able to help these cells stop and reassess the situation. It's the first time this CRISPR activation technology has been used in the 3D brain environment, so we have a lot to learn. I still need to figure out if even if I can get these cells' attention, that will be enough to stop dementia. But for now, I'm excited to contribute how cells speak about dementia to each other in order to better understand how to develop therapies. And I've even been able to find some new definitions of words we thought we already knew. For example, a word that we thought meant divide can actually mean divide and conquer, depending on the circumstances. Understanding this language precisely is going to be key as we begin to develop therapeutics for dementia. But I'm excited to have taken this first step in breaking down this language barrier. Thank you.
Thank you, Amanda. All right, next trivia question. This late 90s dystopian thriller featuring Ethan Hawke and Uma Thurman was the subject of a 2022 Nature Genetics article that discusses the film's prescient forewarnings related to genomic science and technology. Gattaca is the correct answer. Very good. All right. Gattaca, released in 1997. This film presents a vision of a future society driven by eugenics, where potential children are conceived through genetic selection. Ethan Hawke portrays Vincent Freeman, who was conceived outside the eugenics program and struggles to overcome genetic discrimination to realize his dream of going into space. People recognize this? Marin Civic Center, exactly right. This is a special local connection. The exteriors and some interior shots of the Gattaca complex were filmed at the Marin County Civic Center in San Rafael, designed by Frank Lloyd Wright. And what goes around comes around. In 2022, 25 years after the release of the film, Gattaca was screened at the Civic Center as part of its 60th anniversary celebration. That's something I'm sure you'll remember forever. So how are our judges doing? Another little bit of time, okay. I don't know anything more about Gattaca other than what's written here. So it was good film. I think I've seen part of it. Yeah. Quite dysphoric, actually, I think. We good? Okay. All right. Here now for our third speaker, Ayush Srivastav, faculty member Bjorn Schwer. Talk title: Let's Help You Age Better.
Despite all our efforts to stay healthy, there is one condition which none of us can outrun: aging. And I'm not just talking about gray hair or wrinkled skin, but some serious challenges such as diabetes, dementia, Alzheimer's, cancer, the list goes on, all of which are consequences of aging. We could limit the prevalence of all these diseases if we could simply tackle aging. What if I tell you that there is a drug which has the potential to not only extend our lifespan, but also to protect us from these aging-associated diseases? You're going to have a very obvious question now: why are we not taking this drug already then? Well, that's because the drug also has some side effects. While eliminating the harmful processes which are responsible for aging, the drug ends up targeting some important processes also. But just imagine how much we could benefit if we could restrict the effect of the drug to only the harmful processes. Much like, how we want a herbicide to eliminate only the weeds in our garden while leaving the rest of the plants completely unaffected. The problem, however, is that at this point, we don't really know what are these harmful processes that are responsible for aging. And that's because we don't have the tools to study that. This is exactly what my project aims to address: to make a mouse model that would allow us to identify these harmful processes. To do so, I'm going to insert a modified gene in the mouse that would control the effect of the drug in such a way that the drug would only be effective in those parts of the body where the gene is active. In simpler words, I could use this model to limit the effect of the drug to only some specific parts of the body while leaving the rest of the body completely unaffected. And by, let's say, for example, someone comes to me and says that, Ayush, I think if the drug was targeting only the heart and the brain, that would have been the most beautiful therapy. Sure, I could check that with my model. And by doing similar experiments, I would be able to narrow down what parts of the body do we really need to target to ensure an effective and safe anti-aging therapy. This drug, which is known as rapamycin, is the most promising candidate we have so far. And although aging is inevitable, we could use this drug to help us age better. Thank you.
Thank you, Ayush. Okay, while the judges are doing their thing, here's the next question. This famous scientist and science communicator has done The Daily Show, Wait, Wait, Don't Tell Me, Cosmos, and The Big Bang Theory. He also did a postdoc at Princeton University in the early 90s. Who is he? Say again? Neil deGrasse Tyson. Yeah, there you go. Neil deGrasse Tyson. Very good. Astrophysicist Tyson was a postdoc in the Department of Astrophysics at Princeton from 1991 to 94. Today, he's the director of the Hayden Planetarium at the American Museum of Natural History in New York. His interests include extragalactic and intergalactic supernova, dwarf galaxies, stellar populations, and galactic structure. He's also renowned for his work to make science accessible to a wide audience that I'm sure many of us can appreciate through his books, his talk shows, podcasts, and TV series, not to mention an abundance of space-themed neckties shown there. For these efforts, he was awarded the Public Welfare Medal in 2015 by the National Academy of Sciences for his, quote, extraordinary role in exciting the public about the wonders of science, end quote. Less well known, perhaps, is that while a grad student at UT Austin, Tyson was a competitive dancer and won a gold medal with the UT dance team, took first in a national Latin ballroom tournament. Typical physicist. Okay. All right. Judges, we good? Yeah? Okay. Now for our fourth speaker, Helen Ashman, faculty member, Priya Shetty. Talk title, To Treat or Not to Treat, a Balancing Act in TB Prevention. Helen.
Every drug has side effects. Therefore, every time you take a drug, there are benefits and harms. Sorry. Oftentimes, this decision, this trade-off, is entirely up to you. If your blood pressure is too high, you can take a drug to lower it, but to prevent heart attacks. But this can also hurt your kidneys or make you feel dizzy. But what about an infectious... It turns out different people feel different about this decision, and so everyone should make their own decision. But what about an infectious disease? For an infectious disease, your decision also matters for other people's health. And so we need to protect the community and the population at large. As a postdoc at UCSF, I studied the trade-offs around the prevention of tuberculosis, or TB. Worldwide, 1 in 4 people are infected with the TB germs. These people are healthy, but they have sleeping TB germs in their lungs, and these germs can wake up months or years later and cause active TB disease. Luckily, there are treatments that can prevent TB that will kill these sleeping TB germs. Ideally, everyone would take this treatment so we can eliminate TB. So it's important for us to understand under which conditions patients would prefer treatment or no treatment. I use methods from market research to find out. I surveyed people living with HIV in Uganda who are at a high risk of developing TB. Even though 1 in 3 of them will eventually die of TB, only 88% of them were willing to take the old 6-month treatment. But if we offer them the new treatment, the new 3-month treatment, 99% would take it. Good, so we really need to offer that new treatment. By contrast, in the US, most people are at a low risk of developing TB, such as 0.5%. I'm starting another survey to find out whether people at a low risk would also be willing to take preventive treatment. And if so, we can eliminate TB. And is that compatible with individual preferences and values? Every drug has side effects, and we need to help people understand the trade-offs and make the decision that's best for them. Thank you.
Thank you, Helen. All right, the next trivia question. Pterosaurs, prehistoric flying reptiles, made one of their first appearances in a popular film in Arthur Conan Doyle's novel, The Lost World. Okay, this is a little more difficult. What was the approximate wingspan of the largest pterosaur? Yeah, you thought it was all going to be pretty easy, huh? Well, now, you got a hand. Oh, we got a hand up over here. No. but guessing's allowed. 25 feet? Getting closer. A little higher. Nope. Keep we'll give it to 35. 40 feet, 11 to 12 meters. Okay. It was once thought that pterosaurs were not well adapted for active flight and relied largely on gliding and on the wind to stay in the air. However, based on analyses of pterosaur skeletal features, including work done by our own Berkeley Kevin Padian, it's now thought that all but the largest pterosaurs could sustain powered flight. There you go. That's a piece of trivia for sure. Pterosaurs had hollow bones, large brains with well-developed optic lobes, and several crests on their bones to which flight muscles attached. All of this is consistent with powered flapping flight. One of their more recent appearances in pop culture, of course, was in 2001, many years ago, Jurassic Park the 3, although paleontologists noted persistent errors such as depictions of nesting habits and leathery wings that were not updated based on modern research on pterosaurs. That's terrible. How are we doing? You okay? Yeah, okay, onwards. All right. Our next presenter, Kritika Khanna, faculty member John Fahey. Title is Asthma on Fire. Keep calm, it's a false alarm.
Just like a smoke detector in our home, our body has a built-in alarm system. Alarmins are molecules that alert the immune system of any potential threat. For example, when our lungs are exposed to viruses or bacteria, these alarmins get activated and start a cascade of events leading to inflammation, like an alarm that alerts and brings the fire brigade. Now imagine if the smoke detector also goes off when there is just a bit of steam from cooking. Similarly, our body's alarmins can also get activated by harmless things like dust or pollen sometimes. This is a false alarm that leads to inflammation. One important alarmin is TSLP, or thymic stromal lymphopoietin, which is implicated in asthma. In asthma, while all patients have poor lung function, they exhibit diverse features in the clinic. Thus, it is important to find the right treatment for the right patient. Recently, a new therapy that targets TSLP has been developed. However, we are not sure of the ideal asthma patients for this. In my project, I am asking 3 key questions about this alarmin and letting patients help me answer those. First is that which of these patients have that oversensitive detector, and is there a way to identify them easily? For this, I have measured TSLP in the airway fluid of asthma patients and found that about 1/3 of them have high TSLP levels, and these levels are strongly associated with a type of inflammation called type 2 inflammation, where patients have high numbers of white blood cells called eosinophils. Thus, we have identified a subset of patients where TSLP is high, and we know of the markers to identify them easily in the clinic for this therapy. Next, we want to know more about this detector. What are its sources in the lungs? For this, we are carrying out imaging technologies on the lung sections of asthma patients to find the part of the building where the detector is located. And we have narrowed it down to basal cells, which are special lung cells. Finally, we want to look closely at the detector itself. Using RNA sequencing technology, we are trying to, understand the wiring of the detector to see if we can repair it. Overall, in my project, we have identified the ideal asthma patients for this TSLP blocking therapy, and we are exploring the biology of this alarmin to help patients breathe easier and live better. Thank you.
Thank you, Kritika. Okay, here's a question for you. What famous fictional starship's warp core was filmed at the National Ignition Facility, the NIF, at the Lawrence Livermore National Lab? Starship Enterprise? You got it. The USS Enterprise from Star Trek Into Darkness, 2013 film. So the warp core of the Enterprise, as depicted in that film, is actually the NIF's target chamber and several other locations throughout NIF that were also featured in the film. So what actually happens at the NIF? It's called, quote, one of the most ambitious science experiments in history. The NIF uses the power of 192 huge lasers focused on a tiny bead of frozen hydrogen in order to pursue fusion ignition and possible sources of future renewable energy generation. It was opened in 2009, and the NIF achieved fusion ignition for the first time in December of 2022. You may have heard about that. And a little tangential tidbit, Lawrence Livermore National Lab has held their own postdoc research slam since 2021, inspired in part by the example that UCSF has set for the postdoc SAM, which we inaugurated in 2016. So there you go. How are we doing, judges? Okay. Onward. Our sixth speaker is Fate Nuhi. Her mentor is Virginia Sturm. And the title is Life Without Emotions, a Symphony Without Music.
I can feel my face when I'm with you, but I love it, oh, I can feel my face when
I want you to think of a moment in your life when you achieved something amazing. Picture the joy that you felt and consider how it would feel if your closest loved ones didn't react to your happiness, or if you were going through a rough patch and they showed no empathy or care about your pain. This emotional disconnect, while cognition and memory are intact, is like a flutist who plays with precise finger movements, yet neglects the vital act of blowing into the flute. This is what happens in frontotemporal dementia, a form of dementia that leaves the memory intact, but strikes at the very core of our emotional behavior. The way emotions are assessed in clinics heavily relies on subjective reports, which often leads to misdiagnosis of these patients with psychiatric disorders. Beyond misdiagnosis, relying on subjective reports dismisses those who can't accurately express how they feel, or those who are simply unaware of their emotional symptoms. We desperately need an objective, accessible approach that provides the most accurate estimate of emotional well-being. I study patients with frontotemporal dementia because, unlike psychiatric disorders, there are clear, measurable changes in their brain structure that can help us understand what exactly happens in the brain that leads to changes in emotion. Changes that impact our entire body, because emotion functions like a conductor, orchestrating the precise moment each instrument should come into play. From the rhythm of our hearts and flow of our breath to the expression on our faces, they all must harmonize perfectly to generate the appropriate emotional response. We have found that our patients respond to emotional situations with irregular heart rate and facial expressions. Remarkably, the degree of irregularity in facial expression is directly related to the volume of their brains. This means that analyzing the way facial muscles move has the power to serve as a window into the body's internal state to the level of predicting the structure of the brain from the face. Imagine a world where a simple FaceTime with your loved ones can help with early diagnosis of dementia. With this type of metric, we stand at the precipice of a revolution in emotional assessment. Just like measuring and monitoring the blood glucose level in diabetes, we aspire to measure and monitor the emotional behavior to maintain the orchestra in tune and keep the music going. Thank you.
Thank you, Fate. All right, our next interim question here. Inducted posthumously into the National Inventors Hall of Fame in 2014 for co-inventing a frequency hopping communication system in 1942, this inventor also happened to be a glamorous movie star of Hollywood's golden age. You got it. Very good. That's a real trivia question. Good. Yes. Hetty, I don't know. Hedy Lamarr, best known for her work in Hollywood in the 1930s and 40s, starring films such as Ziegfeld Girl and Samson Delilah, but she also worked with Hollywood composer George Antel to invent a frequency hopping technique that today is referenced as an important development in the field of wireless communications. Born in Vienna, Austria, Lamarr had at one time been married to a munitions manufacturer, giving her the foundation for her knowledge of weapon systems, including torpedo control systems. And Lamarr, man, this stuff, what you guys come up with. Lamarr and Antil's frequency hopping reduced the risk of detection or jamming of radio-controlled torpedoes. And although they never profited from their invention during their lifetime, it was acknowledged by the Electronic Frontier Foundation in 1997 as an important development in wireless communications. And in addition to her place in the Inventors Hall of Fame, Lamarr also had a star, she also has a star on the Hollywood Walk of Fame, bestowed in 1960, and rumor has it that Lamarr was one of the inspirations for the original Catwoman of the Batman comic. There, that'll stick. Okay, judges, we good? All right, our next speaker, Biafra Ahananu. Faculty member is Alan Bassbaum, and the title is The Path to Pain. It's Not Only in the Brain.
Dance. Did I mention I like to dance? By the way, I love to dance. Quick note, I need to dance, 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 dance. Did I mention I like to dance?
So many people like to dance and do other activities like playing soccer, and yet many are unable to, in part due to ongoing pain. Now, pain has many causes. It can either be from external injuries, such as pain to the body, or from internal changes, as in the case of autoimmune disorders or diabetes. And we would want to change, but while many treatments exist, painkillers can often be ineffective in certain patients or have unacceptable side effects, as we've seen in the opioid epidemic. And so we would like to change this, in part by better understanding both the neural and molecular mechanisms of pain. Now, pain is critical for survival. You need it to escape from dangerous stimuli or to attend to an injured body part. And yet we don't always perceive pain in the same way. And so here you're seeing the last minute of the last match of the season, and the team must score in order to win. And right as he's about to go into the goal, you see that a defender goes in and stamps on his foot. And while many of us will think that soccer players kind of, fall over a lot, exaggerate their injuries, this is painful, and yet he ignores it, goes on to score, and they win the league in dramatic fashion. And so what we want to do is understand where along this pain pathway these stimuli are being blocked, and can this tell us about what's going on in chronic pain where we think that incorrect stimuli are being sent to the brain. So how are we going to do this? So we need to be able to monitor neural activity in awake animals, in part to relate neural activity to perception. And while me and others have done this in the brain, we want to look at the spinal cord to better understand how is that signal being processed before it even gets to the brain. And so to do that, a colleague and I developed both experimental and surgical approaches that have allowed us to peer into the spinal cord over time. And we do that by expressing a protein in neurons that fluoresces or blinks like a flashlight, and then we can go in and peer at it with a microscope. And so what this allowed me to do is go in and peer into the spinal cord over time and look at the changes that are going on. And so what I've been able to do is use both computational approaches I've developed and new techniques to see the same neurons are being activated day in, day out as the animal responds to different types of painful stimuli. And so using this approach, we're now seeing in pain models, do you get an increase in the neural activity as chronic pain develops? And using new painkillers being developed here at UCSF, do you also now see that activity going back to normal, suggesting that these painkillers will be helpful in alleviating pain in patients so they can go back to doing activities that they really love, like soccer or dancing. Thank you.
Thank you, Biafra. Okay, this next one's close to home. UCSF's Genentech Hall was in costume as Mendel Hall in what film? You got it. Very good. All right. Yes, so Elliot Gould played the fictional UCSF professor Ian Sussman, and this building we're in here now played fictional Mendel Hall in Steven Soderbergh's 2011 film about the global spread of a highly contagious virus. Genentech Hall was not the only San Francisco location to appear in the movie. Filming also took place in Golden Gate Park and Chinatown and Candlestick Park, among other sites. And as you, I'm sure, remember, the film had a resurgence in popularity in 2020 for rather distressing reasons. And in fact, with the onset of the very real and of course, serious pandemic, Contagion became the 7th most popular film on iTunes and shot up to high rankings on other streaming services. In reference to its surge in popularity, screenwriter Scott Burns told the Washington Post, quote, "People still don't seem to grasp the situation we're now in and how it could have been avoided by properly funding the science around all of this. I had access to great expertise, so if people find the movie to be accurate, it should give them confidence in the public health experts who are out there right now trying to guide us." There you go. Judges? Yep, okay. All right, now for our 8th speaker, Laura Pershan. Faculty member is Noelle Latoile, and the talk title is Wormnado, What a Tiny Worm Can Teach Us About Collective Behavior. Laura.
You spin me right round, baby, right round, like a record, baby, right round, round, round. You spin me right round, baby, right.
Everyone here is an expert at collective behavior. We do it every day. At the grocery store, we sort ourselves into checkout lines. In city traffic, we merge and yield to accommodate another UPS truck stopped in the road. Each of these behaviors requires that we perceive other individuals and then modify our own behavior in response. And while this might feel second nature, it's actually a pretty complicated task. Luckily, we have 86 billion neurons to help us. And in fact, it's thought that social species evolved larger brains just to handle these types of challenges. So you can imagine my surprise when I started to notice that the tiny worm we work with, which has 300 neurons, would sometimes spontaneously self-organize and execute a choreography more advanced than the average high school marching band. Here you see 2 dishes of worms. Now imagine for a moment that you've always seen dishes that look like this left image. Then you notice the dish on the right. And in this hypothetical, it's late at night, you've been staring at a lot of worm plates. So you're like, hang on, is it just me, or do these worms look kind of organized? Are these guys up to something? Well, to summarize months of subsequent work, they are up to something. It turns out that in response to a lid going on the dish, an entire population will simultaneously orient themselves parallel to the edge of the dish and then migrate in a bidirectional spiral to the center. Now, these worms can't see or hear, and yet they're communicating because the more worms you have in the dish, the better they get at organizing. So at this point, I turned to Google. I did some weird searches, would not recommend. Worm vortex, possessed worms spiraling. I did find that Rihanna's backup dancers once did something pretty similar, but the internet confirmed what I already suspected, that these worms aren't supposed to be talented backup dancers. So I called it the Wormnado and decided I had to study it. But why should we care how these worms nado? What could I possibly tell my relatives? Well, we've seen that collective behaviors often apply across systems. We have models for machine learning based on ant behavior, and immune cells can swarm just like insects. So the parts might be different, but the principles are reused. Ultimately, though, I love this project because it could only have happened by accident. And so it reminds us that doing science is about being curious and open-minded. It's about paying attention when nature does something surprising, because that's where we find truly novel biology. Thank you.
Thank you, Laura. Okay, here's one for you. Bunny tails are not the whole story. This beloved author was also an accomplished naturalist and botanical illustrator. Oh, I guess you've already. Oh, yeah. All right. No more swag for you. There you go. That's exactly right. Okay. I'm very excited back there. Good for you. Beatrix Potter, British author, was author and illustrator of 23 children's books, including The Tale of Peter Rabbit, of course, which she self-published in 1901. In the years since, her books featuring various animal characters have sold more than 250 million copies. Potter also had a lifelong interest in the natural sciences and in scientific illustration with a particular focus on mycology. She painted her first known watercolors of fungi at 20 years old and completed over 350 throughout her life. She died in 1943, but in 1967, mycologist Finley, including many of her fungus drawings in his book, Wayside and... Oh my God, I can't believe we get into this stuff. Sorry guys, you have done a fabulous job, but who knew there was a book called Wayside and Woodland Fungi? Okay, so I'm sure I've hurt the feelings of some mycologists in the room. Realizing Potter's dream of having her fungi paintings published. Biographer Linda Lear wrote that Potter, quote, never saw art and science as mutually exclusive activities, but recorded what she saw in nature primarily to evoke an aesthetic response. However, she also noted that her, quote, watercolors are considered so accurate that modern mycologists refer to them still to identifying fungi. So that's serious business. Good for her. Okay, we all set? Okay, our penultimate speaker, Christopher Williams. Faculty member is Atul Butte, and his talk is Code Blue, Unmasking Racial Disparities in Emergency Triage. Christopher.
When you go to see your doctor for a health problem, you expect to be treated in the same way, regardless of the colour of your skin. Yet, we live in an inherently biased society where examples of systemic racism often make headline news. Biased policing strategies, residential segregation, unfair lending practices are but a few examples of this. I remember when I first saw some of these headlines, I was left thinking, what other areas of my life may be affected by structural racism, and what can I do about this? Here at UCSF, I investigate whether this bias exists within our own healthcare system. I focus my attention on the emergency department, which is typically the first place people go to when they have a serious medical problem, and the origin of many of the downstream decisions and treatments that patients receive. Consequently, it represents an ideal place in which to start looking for racial disparities in healthcare. When patients come into the emergency department, they are first triaged. They will have their blood pressure, heart rate, and other vital signs measured, and they are given a score that dictates how urgently they should be seen. This is known as the acuity score. In my research, I investigate whether racial disparities exist in the assignment of acuity scores during the emergency department triage process. To do this, I leveraged an unprecedented dataset of 350,000 patients presenting to the emergency department. I used an artificial intelligence technique called natural language processing to cluster patient symptoms so that a fair comparison of patients presenting with different types of conditions could be made. I additionally extracted a host of clinical and demographic details relating to each emergency department visit. This was a hugely important step to ensure that all confounding variables are suitably accounted for. I then created statistical models to examine whether Black, Hispanic, and Asian patients are more likely to receive lower acuity scores than white patients. What I discovered was both illuminating and disheartening. The data revealed that patients from racial and ethnic minority groups were significantly more likely to receive lower acuity scores and be seen less urgently compared to their white counterparts. This finding persisted regardless of various patient characteristics, including patients' age, sex, vital signs, and presenting symptoms. I plan to use these findings to delve deeper into the root causes of racial disparities. Where else in a patient's care do these disparities exist, and how do we develop effective strategies to tackle this inequity? By doing this, it is my hope that, together, we can work towards a future where everyone can go to see their doctor and be treated in the same way. Thank you.
Thank you, Christopher. Okay, this is actually our last trivia question. and that is Adam Summers, then a postdoc studying fish biomechanics at UC Berkeley, advised filmmakers on fish locomotion, how fish scales reflect light, and the mechanics of waves during the production of this 2003 animated Disney film. That's exactly right, Finding Nemo. Good for you. All right.
Somewhere beyond the sea.
Finding Nemo won the Academy Award for the Best Animated Feature in 2003, as well as several other awards. Dr. Summers is now on the faculty of the School of Aquatic and Fisheries Sciences at the University of Washington, where his research focuses on understanding the effects of material properties on the form and function of organisms. Summers has been active in science communications and writes on his lab website, quote, "Communicating the research we do in the lab is important. Communicating science at large is more important. Everyone in the lab takes this job seriously with the proviso that it has to be fun and if it's going to get done." So good for you on Finding Nemo. How are we doing, judges? All okay. And finally, our final speaker, Jay Maltzo Anderson. Faculty mentor is Susan Lynch, and title is "This Stool is Taken: Harnessing the Power of Poop."
Poo, 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 poo in the loo-loo. Poo, 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 poo. Everyone needs to poo.
Looking around this room, I can tell you that we all share one thing in common. And while you might think it's high rent, it's a little more personal than that. I'm talking about poop. Poop has a bad reputation. The end waste product of our digestion, most of us wouldn't think twice about flushing it down the toilet. But poop is amazing, and it has a lot of beneficial properties that we can use to help treat and cure diseases. Specifically, it can be used to help benefit patients with ulcerative colitis, a chronic inflammatory condition of the gastrointestinal tract. Treatments are available, but they carry risks, are very costly, and patients often develop resistance over time. In advanced cases, surgical intervention may be required to remove part of the bowel, and concerningly, the rates of UC are increasing globally. One treatment that has shown a lot of promise is the fecal microbial transplant, or FMT. This is where the poop from a healthy donor is transplanted into a patient. But while FMTs are very effective for UC, they don't work for everyone. They also carry risks, and they require multiple administrations. So, what to do? So to help treat these patients, I'm trying to replace the FMT altogether with a live microbial biotherapeutic, or as I like to call it, poop pills. I'm harnessing the good products from poop, and I'm using them to create pills to help treat UC. To do this, I've leveraged a clinical trial here at UCSF, where UC patients were administered FMTs. By taking poop from patients who positively responded to these treatments, I was able to isolate and culture their beneficial bacteria. I then sequenced these bacteria to find the genes responsible for producing key metabolites, or small molecules, that decrease gut inflammation and led to disease remission in these patients. Ultimately, these beneficial components will be used to make up these poop pills, which will then go on for clinical trials as a safer and more effective therapy for UC. So the next time you flush the toilet, I hope you don't think about me, but rather, I hope you think about how poop will help benefit these patients as a better therapy. Thank you.
Okay, so that's our 10 presenters. Actually, if you don't mind, the finalists, if you could just come down here, for all of you, all 10 of you can just come down here for a moment, please. Come on down. And yes. That's right. So as I said at the beginning, these are 10 incredible postdocs who represent more than 1,000 postdocs here at UCSF. And both personally and I think everyone here in the room, we cannot thank you and all the other postdocs for the work that you're doing on behalf of the people that we serve. So let's give a final round of applause for everybody. Okay, you guys can go back, and they're in a very relaxed state now, you can see. Okay, so just a couple other things. This is now our chance as the audience to play judge today. So vote for your favorite talk to win the People's Choice Award by going to the URL or the QR code that will be displayed on the slide momentarily. There it is. Those of you viewing remotely, you'll also see this link right under the video in your browser or in the description area below the video on YouTube. And take this serious. This is really important. Like, you guys get to vote for this particular award. Take a minute to think about it, and then please submit your vote soon. Audience voting will be open for just about 5 minutes or so. And now, while the judges are whisked away to a top secret location to confer and decide on today's winners, please take a short break and stretch your legs. We'll have some light refreshments for you outside in the atrium, but you'll definitely want to return at about, 5:25 or so, which is in 15 minutes. We'll all reconvene here to announce the winners, and of course you can watch the announcement remotely too. Okay, so have some fun. We'll be back shortly. Thank you.
I have the envelopes, and just to torture you just a tad bit more, I'm gonna say some thank yous before I present the envelope. So this event does not happen without some serious choreography, and a lot of people have been working behind the scenes to pull this off. So I just want to say, first of all, thanks to our MC, Dan Lowenstein. There are 2 rounds to get to the finalists. So we had screening judges. Their names are on your program, so I won't call them. They're all there. All their names are on their program. We have Postdoc Slam coaches. We have the Office of Career and Professional Development that facilitated the coaching. We have our ETS guys who are all in charge of live stream. Campus Life Services, let me see those of you who won something today. Yes, you're still here, awesome. Our Graduate Division comms team for getting the word out, and the Graduate Division and Student Academic Affairs, lots of them around helping out. So join me in saying thank you to all of them. I do this at every slam, and it's no less heartfelt. Folks, there's a young man in this room that puts all this thing together. He creates all those trivia questions. He literally does everything. This man eats, sleeps, and dreams slam. It is a labor of love for him, and he's always trying to hide. But Eric, stand up so that we can appreciate you. All right, without much further ado, we have 4 winners. Ladies and gentlemen, the presenter for the People's Award is none other than Won Ha.
All right, before we actually get to announcing any of the winners, let's give a big round of applause to all of the contestants. I don't want to reveal too much about what happens in the green room, but there were a couple of fights that we had to break up among the judges. This was a very tough decision. All right, so I'm here to announce the People's Choice Award. I get to open the envelope and act surprised, even though, I know who this is. So after a tally of 400 141 votes. And introducing a new word to the UCSF lexicon, wormnado. The People's Choice Award goes to Laura Person. Congratulations, Laura. Thank you. And we're supposed to take a photo over here.
All right, that is not your money. Your money will magically appear in your account at some point in the very near future. Thank you, technology. All right, our presenter for third place winner is our high school teacher, Curtis Chun.
Thank you again. Excellent job, everyone. As a high school teacher, I watch a lot of these presentations, and you guys were great, all of you. So we narrowed it down. It was a tough decision, and we got down to the top 3. And so your third place winner, well, I'll just hand this to you later, with a presentation that was important and hopefully impactful on unmasking racial disparities in emergency triage is Chris Williams.
Chris just made $1,000. Not bad. Not a bad day's work. All right. And now the presenter for the second place award is Audra Johnson.
Thank you very much. Hopefully, I could take an elbow here, bring it down. So second, everybody likes a height joke, right? Let's get to it. I'm gonna stand up, okay? I don't have that limber postdoc back anymore. So for our second place winner today, very excited to announce this is Kritika Khanna.
Kritika just earned $2,000. Yes. Fly anywhere. Ladies and gentlemen, the first place award will be presented by our own Helen Wilsey. Come on up. And just to remind you, the first place award is $4,000.
It is my great honor to present this first place prize. If you had been in the judges' corner, you would have heard us all talk about both the amazing humor, but also the phenomenal perspective on a life in science and how fun it is to look in your dish and see something weird. Laura Person, Wormnado.
No, I don't. I'm good. All right. Thank you all so much. Can we just give all 10 contestants another round of applause? All right. Thank you all so much for sharing this time with us. We appreciate it. Go finish the refreshments outside. Have a chat with each other, mingle and network. Thanks, everyone.